Hi. In this video, I will be talking about how DNA bends, folds, compresses, and stretches in order to maintain the structure's strength and ensure it takes up as little space as possible. Many of the ways DNA can be packaged can also be exploited for DNA regulation, but this video will only cover the packaging itself, not how it regulates the DNA. That will be in other videos. This is an overview of what we will be covering today. I will only be giving a brief introduction into each of these things. I don't want this to be extremely in-depth. The purpose of this video is to just give you an idea or a bigger picture of what DNA and chromosomes actually are, because we initially learn them sort of abstractly as inheritable bits of information, and we can sometimes forget that they can take up whole 3D structures. There are so many labs that strictly focus on how DNA is packaged just because it's so complex. But anyway, I just wanted to clarify from the beginning that this video is only scratching the surface of what this field of study has discovered, and who knows, maybe you'll find it interesting enough to look into yourself or even make a career out of. So this is the most famous structure of DNA, also known as the canonical DNA. There are other conformations of DNA with various structures, but that will not be the focus of the video. I'll probably make a video about them though. This video is really more about the different techniques DNA uses in order to exist in a very small volume in the nucleus, and these other structures do not really have an effect on the amount of space the DNA takes up. The term duplex means that there are two strands of DNA, so if this picture had 3 strands, it would be a triplex. That also exists, but it will also be talked about in another video. Along with the term canonical DNA, this is also known as naked DNA since it is not accompanied by other proteins. Prokaryotes are exclusively known to have this type of DNA, so from here on out I will be talking about eukaryotes. Also, just a warning, there's a certain Australian magazine called DNA that has some explicit images, so I wouldn't go too far down the images tab if you are searching up naked DNA on Google. This is a picture of one strand of DNA. DNA is negatively charged due to the phosphate group in the DNA backbone. These are proteins called histones, and I will talk more about them in a second. Since the histones are made up of a lot of positively charged amino acid residues, that being lysine and arginine, they attract the negatively charged DNA. In total, a single nucleosome consists of the core DNA wrapped around 8 histones called an octamer. Each nucleosome is joined together with linker DNA whose length fluctuates, so there could be some nucleosomes with a little space between them and others with a lot of space between them. Then a 9th histone protein called H1 acts as a sort of clamp, and you can see that it decreases the diameter from 11 nanometers to 6 nanometers, further minimizing the space DNA takes up. In reality, all the types of histones like H1, H2, H3, and so on are protein families, meaning that there are many proteins that can act as an H1 protein. So when I say the H1 protein, I really mean a H1 protein, so just keep that in mind. H1 is also called the linker histone, or H5, but I'm just going to stick with H1 since that is what the diagram says. Once the H1 histone protein binds to the nucleosome, it becomes a chromatosome. You might have noticed that the chromatosome contains a much longer portion of DNA than the nucleosome, and this is because the H1 interacts with some of the linker DNA, about 10 base pairs on each side, so 20 in total. The main distinction between nucleosomes and chromatosomes came from experimental research shown in this diagram. It looks a bit complicated, but just focus on these parts. If you compare the denaturing process of the chromatosome and the nucleosome, you can see that the H1 protein protects these extra 20 base pairs from the denaturing process, where the DNA from the nucleosome is 145 base pairs long, while the DNA from the chromatosome is 165 base pairs long. Without the H1 protein, the DNA wraps around the nucleosome a total of 1.65 times. Meanwhile, when the H1 protein is present, the extra 20 base pairs and the tighter intermolecular forces allow the DNA to be wrapped around the chromatosome a total of 2 times. This is a bit confusing, so I made a table. The difference between chromatosomes and nucleosomes is, in my opinion, kind of redundant, but you may encounter this in the future. That being said, if you get them mixed up, don't feel bad, because there are many research articles that I saw that also got these confused. And I don't just mean calling the chromatosomes nucleosomes, because nucleosomes is a much more popular term, but I mean actually mixing them up. I'm just going to call them nucleosomes for the rest of the video though, because that's what most people do anyway. If you do want to memorize this table, just be careful with remembering that DNA wraps around the octamer 1.65 times in the nucleosome, but is around 165 base pairs long in a chromatosome. It's very easy to mix those two up because it's essentially the same number, just with a different decimal place. Before we go further into how DNA condenses in the nucleus, I want to talk about the difference between heterochromatin and euchromatin, since the other structures I will be talking about are dependent on this distinction. The main difference between them is that heterochromatin is less transcriptionally active, while euchromatin is more transcriptionally active. There are many ways in which this occurs, and I have a table of just a few of those ways. You shouldn't view heterochromatin or euchromatin as having explicit conformations. It is just a way for people to categorize more condensed structures with less condensed structures. The H1 proteins, if you remember from the last slide, increase how tightly the DNA is wrapped around the nucleosome, so it is harder for transcription factors to bind to the more tightly packaged DNA. If you don't know what transcription factors are, I will talk about them more in another video, but essentially they are proteins that transcribe DNA into RNA. As for methylation and acetylation, those have to do with epigenetics and how different markers bind to the specific histone tails to control gene expression. The reason why I put both methylated and demethylated and acetylated and deacetylated is because I want you to understand that this is all relative. Just because a gene may have a histone with a methyl group doesn't mean that that gene is downregulated. All that matters is if it has more or less methyl groups than at other times. This also introduces the idea of chromatin changing from heterochromatin to euchromatin. So there's a further classification that I want to add. Heterochromatin can either be constitutive or facultative. Constitutive heterochromatin is always minimally expressed, while facultative heterochromatin can change from euchromatin to heterochromatin. I put etc. at the bottom of the table because this is basically all that we have covered so far, but at the end of the video I will show the full list that includes all the different trends that can be seen in heterochromatin and euchromatin. So every eukaryotic cell has nucleosomes, but this is where there is a fork in the road. The possible configurations beyond this point are known as higher order packaging and are dependent on cell type, the state of differentiation the cell is in, and environmental factors such as salt concentration. This picture of many nucleosomes in a sequence is called chromatin and is about 10 nanometers in width. I won't go too much into this because it has more to do with DNA regulation, but the length of linker DNA between the nucleosomes can be increased or decreased. The less linker DNA and the more tightly packed the nucleosomes are, the less likely a regulatory protein will bind to the DNA because they are blocked by adjacent nucleosomes. One common form of this bunching up is called clutches. These clutches can be small or large. The large ones are extremely difficult for regulation proteins to access, while the smaller ones are a bit easier. The small ones are more commonly found in stem cells since they haven't differentiated yet, and the large ones are more commonly found in somatic cells that have differentiated. This makes intuitive sense if you recognize that the larger clutches are more restrictive on gene expression. And again, many factors influence the clutch size. To relate this back to the heterochromatin and euchromatin, it can be said that the larger clutches form less transcriptionally active areas, meaning it would be considered heterochromatin, and the smaller clutches form more transcriptionally active areas, meaning it would be considered euchromatin. In the scientific literature, the linker DNA is also commonly called nucleosome depleted regions, or NDRs, and they're usually where transcription factors are. This is a picture of what used to be the accepted higher order structure for most eukaryotic heterochromatin. This is called the solenoid structure, and although it, as well as its related alternative models such as the zigzag model, have been basically abandoned, I want to talk about it because its abandonment was so recent, and some textbooks and maybe even your class might still have this in their curriculum. But just know in the back of your mind that this does not appear in most circumstances. The solenoid model appears when the nucleosome interacts and associates with its adjacent nucleosomes, so they're neighbors. However, in the zigzag model, I know it's kind of hard to see, but each nucleosome would interact and associate with every other nucleosome. For example, if I labeled them all in a sequence, all the odd numbered nucleosomes would interact with each other, and all the even numbered nucleosomes would interact with each other. As with many of the previously mentioned packaging methods, this is dependent on a variety of factors. Both structures are considered heterochromatin and are formed when the 10 nanometer fiber of chromatin coils up into a 30 nanometer fiber. When it was first discovered in vitro, so in a lab, it was demonstrated that the structure could only exist in the presence of the H1 protein. Since we had not discovered a biological purpose for the H1 protein in regard to DNA packaging up until that point, and the solenoid structure required it for the structure to even take conformation, it made sense to assume that this was how DNA was packaged. Additionally, if you restricted yourself to the 10 nanometer fiber, you would not be able to fit the long length of DNA inside the nucleus, but with the 30 nanometer fiber, you could. So everything pointed towards the solenoid structure being a vital component for higher order DNA packaging. There were even experiments done regarding how the solenoid structure is unwound before the DNA can be transcripted into mRNA, or before the DNA is replicated so mitosis can occur. However, as technology improved and as more imaging experiments were done, many studies tried to observe this structure in vivo, so in actual living organisms, and it was only ever found in vivo in a few studies. Some examples include a chicken erythrocyte and in a starfish spermatozoid. Nevertheless, the model was wrong, or at least not generally applicable in most scenarios. So what actually goes on? To explain this, I think it is easier for me to zoom all the way out and then get smaller and smaller until we get back to the stage of compaction, the nucleosome clutches. This is how most of the research in this field has been done. There is one group focused mainly on bottom-up strategies, and another group focused mainly on top-down strategies. Where they meet in the middle is where we currently are, so let's see what the top-down group found, and then we can bridge the gap between the two. As you probably know, we have many chromosomes in our nucleus. During interphase, the inside of the nucleus looks like this. Obviously, the chromosomes aren't actually colored like that. The image is just showing you that they have their own designated areas that seem to never tangle up. These areas are known as chromosome territories. The reason for why they're chromosome territories makes intuitive sense. The chromosomes have to be untangled for mitosis to occur, and that can't happen when all the chromosomes are tangled up in the same territory. But how does this actually work? Well, it works through loop extrusions. Loop extrusions occur at the chromatin level, so it is kind of hard to conceptualize how a process at the chromatin level can prevent entire chromosomes from getting tangled, but hopefully after a few slides this will make sense. This is an electron micrograph of a metaphase chromosome, with many of the additional proteins that condense DNA being stripped from the DNA, so it all spills out from the chromosome protein scaffold, which is another way for DNA to condense and take the shape of a chromosome. This is new information, the fact that there are what's called non-histone proteins in the nucleus that also help package DNA. Non-histone proteins is actually a large umbrella term that also includes literally every other protein besides histones, so transcription factors, enzymes, and a whole bunch that have nothing to do with DNA packaging. But chromosome scaffolding is one of them, and it does deal with DNA packaging. Proteins that were stripped off the DNA to make this image also aid in condensing the chromatin into a chromosome. You probably notice that this image distinguishes between DNA loops and DNA that is anchored to the scaffold. if you just visually compare them, you can see that the DNA loops are extremely exposed compared to the DNA that is anchored to the chromosome scaffold. I'm just pointing this out to reinforce the idea of heterochromatin versus euchromatin. The DNA loops are euchromatin because they are more accessible, while the DNA anchored to the scaffold is heterochromatin because they are less accessible. Now, to explain these loops, I'm going to first talk about a method of keeping sister chromatids together, and then I'm going to compare that to how chromatin inside a sister chromatid stays together. Don't worry if you completely forgot how mitosis works, because I'm going to give a brief overview of it right now. First, at least in humans, the cell has 46 chromosomes, 23 from mom and 23 from dad. In the synthesis phase, the S phase of the cell cycle, DNA is duplicated, and now each chromosome that looked like the picture on the left now has a sister chromatid, and it looks like the picture on the right. This is kind of confusing because biologists define a chromosome by the presence of a centromere and not the amount of sister chromatids there are. Since both of these chromatids share a single centromere, they both are considered a single chromosome despite the clear difference of duplicated DNA post-S phase. It is also confusing because the picture on the right is what people normally think of as a chromosome, but in reality, it is known as the mitotic chromosome because it only looks like this during mitosis. The rest of the cell life and the majority of the cell cycle, the chromosome looks like the picture on the left. It is also important to note that the centromere is not a structure, but a region denoting the center of the chromosome. So when the sister chromatids split during mitosis, each of them has their own centromere. What's important for this video is how sister chromatids stay together. They are attached at the centromere, but also if we zoom into this section, that shows both chromatids. They are attached all along the chromosome lengthwise with a ring-like protein complex known as cohesin. The chromatin from one sister chromatid loops through the cohesin, and the chromatin from the other sister chromatid loops through the same cohesin protein complex, linking the sister chromatids together and preventing them from separating from each other. This is not what makes the loops that we saw in the black and white image. To explain those loops, we have to zoom into a single chromatid where we can find a similar protein complex known as condensin. So you can imagine, instead of two strands of chromatin in the loop created by cohesin, one strand of chromatin is used for condensin. The way it works is the condensin protein complex comes along and attaches to the single strand of chromatin. The chromatin gets pulled through the condensin protein complex until it is stopped by another protein, called CTCF. As enough of these happen, the chromatin becomes more and more untangled. The way I remember the difference between cohesin and condensin is that the purpose of condensin is to condense the chromatin, while if you remember cohesion and adhesion in water molecules, cohesion is water's ability to stick to each other. So cohesion enables sister chromatids to stick to each other. Okay, let's zoom back out. Going down one level of organization from the chromosome, regions of the chromosome can be categorized into smaller regions that are simply called compartment A and compartment B. Each chromosome can have a multitude of these compartments, but they usually fall into either category. Compartment A is similar to euchromatin in the sense that it represents areas that are heavily transcribed, and compartment B is similar to heterochromatin since it contains a lot of DNA that is not heavily transcribed. Another similarity is that they can also be described as constitutive and facultative. Constitutive meaning its purpose is sort of set in stone and it cannot change transcription level, and facultative meaning that it can fluctuate between being transcribed a lot to not as much. Usually A compartments are found near the center of the nucleus, and B compartments are usually found on the edge of the nucleus. The main hypothesis for this trend is that the center has a high concentration of transcription-related proteins and other structures such as nuclear speckles, which are organelles inside the nucleus that increase gene expression to genes in a close proximity. So inside each compartment, there are even more distinct regions known as topologically associated domains, or TADs, that separate sections of chromatin from each other. similar to how the solenoid and zigzag structures formed because of nucleosomes that associated with each other. The interactions between the chromatin define the region that the TADs take up. The boundaries between TADs also define where one TAD ends and another starts. The word topological is there to reinforce the idea that this is all happening physically. There are loops and fibers that interact with each other to control gene expression, and they are all found in a distinct domain. You might have heard about enhancers needing to be physically close to promoters, even though they are many base pairs apart. This is one of the ways that this happens, and it is all mediated by loop extrusion. There have also been experiments that showed that when the boundaries of TADs are broken, enhancers from one TAD change gene expression in another TAD. So it is also a way of restricting gene expression in other areas of the chromosome. If you don't know what enhancers and promoters are, just know that the loops from the loop extrusion bring in regulatory elements closer to the genes they control. Okay, so right now we have introduced essentially every method of DNA packaging except for one. This middle question mark is a structure that bridges the gap between nucleosome clutches and topologically associated domains. It is known as chromatin nanodomains. Yep, it's another domain. It's also abbreviated as CNDs, but that's not a pronounceable acronym, so it's just chromatin nanodomains. Just like how inside each chromosome there are many A and B compartments, and inside each A and B compartment there are many topologically associated domains, there are also many chromatin nanodomains inside each topologically associated domain. This one is not too difficult to understand. It's just what it looks like in the picture. It's just larger bunches of nucleosome clutches. Alright, that's about it. As promised, here's the full table of things that can constitute heterochromatin and euchromatin. Thank you for watching, and if you have any questions, feel free to leave them in the comments and I will try to answer them to the best of my ability. Have a good day.